Okay, this is the second half of chapter 17, talking about gene expression control, and now we're going to talk about it at the level of RNA. So we're going to talk about RNA splicing, RNA degradation, and RNA interference. So we've already talked about alternative splicing and the spliceosome. So I want to add a little bit of detail to this and let you know that the reason we can have alternative splicing happen is because there are sequences in the RNA that are recognized by proteins, and you're going to see two main types called SR proteins and HNRNP proteins. These are just general classes. So sequences in the RNA that are recognized by proteins that recruit or block the spliceosome. Okay, so just like when we talked about transcription, you have basal transcription factors that are common for all genes, or all RNA Pol II genes. And then you have the regulatory proteins, the enhancers, the repressors that can affect gene expression. Similarly, with alternative splicing, the spliceosome is the general set of proteins that work to do the splicing, but the SR proteins and the HNRP proteins and other proteins are the ones that actually direct the splicing, or I'm sorry, the spliceosome where to go. So you can see these little sequences here. So you have ESE, you will have ESS, not shown here, ISE, also not shown, and ISS. So these are all RNA sequences. Okay. The E, I'm going to write over here, E stands for exon, the first E, and I stands for intron. So you can have exonic sequences and intronic sequences. So that tells you where the sequence is located, in the exon or the intron. The S stands for splicing. Okay, so these are all splicing sequences. And then the second E or S, these guys, I'll write it up here. This E stands for enhancer, and the S stands for suppressor. Okay, so let's get this a little organized. So up here, ESE is an exon splicing enhancer. Okay, so what that's doing and what it's showing you here is that in this case, the SR protein is binding the exon splicing enhancer, and it is promoting U1, part of the spliceosome, to cleave here, and blocking it from recognizing this GU sequence. So remember that there's consensus sequences at the 5' and 3' ends of the introns that help the spliceosome recognize where to splice. But you can imagine these sequences aren't super long, so you could have multiple of these sequences in introns and exons. And even though we draw them like box and line, it's really one long mRNA. So the proteins can't recognize this is an exon, this is an intron, except by sequence. So you need some additional proteins to direct the spliceosome exactly where to work. This is a different example. So here's an exon splicing enhancer, and it is recruiting the U1 to the 5' splice site of the intron, and then the U2 UAF to the 3' site of this intron. So this is really intron 1, and this is, say, intron 2 surrounding this exon. So these SR proteins will bind these enhancer regions and really direct the spliceosome where to bind. Okay. You can also have, over here we have an intron suppressor, so an intron splicing suppressor sequence. Okay. And when the SR protein binds here, it blocks the spliceosome from recognizing the 5' and 3' splice sites, and so this intron may actually be included in the alternative splice. And remember, I told you, yes, we usually say exons, alternative splicing, but it's way more complicated than that. So I want you to understand that there are sequences that interact with proteins that are directing the spliceosome as to where to splice. This is just another example. Here we're bringing in the hnRNP. So the SR proteins is a general class. So this is the big name. And they are proteins with serine, arginine, rich amino acids. So with serine and arginine, we'll just say amino acids. That's what the S and the R stand for, serine and arginine. So they have RNA binding domains. They recognize specific sequences. So there are going to be lots of different SR proteins expressed. Okay, so these are not general. We name them SR, but when you got into a specific gene, they would actually have very specific names. HNRNP just stands for heterogeneous, it's a really indescript name, heterogeneous ribonucleo, it's not going to fit when I say this. Ribonucleo protein. Okay, so it's again a general class. You would have specific ones. Ribonucleo tells you it's made up of RNA and protein, and it hangs out in the nucleus, which is where alternative splicing happens. Many HNRP proteins are inhibitory of alternative splicing. So you don't need to know specifics. What I want you to understand, let's look at the next slide. Okay, maybe. What I want you to understand is that you can have multiple sequences. So this is just kind of a zoom in. Here's a list of all these different proteins that can bind these exon splicing enhancer and silencing or suppressor sequences. Sorry. So these are all the proteins. And depending upon which proteins you have expressed will depend on which exons, or parts of the exon in this case, are included or excluded. So remember, we're talking about, again, layers. So you have all these sequences. The sequences that are used depends on the proteins that the cell expresses. We talked about the spinal motor neuron, SMN1 and 2 genes, and I showed you that when you get a mutation in gene 1, it unfortunately makes a very unstable protein like the one normally expressed from SNM2 because it doesn't have exon 7, and that causes a genetic disease where the muscles don't fully develop and the person is wheelchair bound. So I wanted to show you what's really happening there. And what's happening is that you get this mutation. Oh, here we go. In exon 7, a change of a C to a U. And that is in the that is in the, where am I looking here? Sorry. Oh, here we go. I lost my sight here. So let's zoom in here. That one, excuse me, that C to U nucleotide change in the ESE, this one right here, this gray one, changes it to an exon splice from an enhancer to a suppressor. That means that you are not going to include exon 7, and that makes an unstable protein. So remember, normally we have both of these genes and the SNM1 makes the stable protein, but if you have the C to U change, SNM1 becomes just like SMN2, and you no longer have a stable protein made. Okay. So again, this is all based, now I understand I have a page out of order here. It's messing me up. Hold on just a sec. So the take home from all of these is that you have gene sequence regulators that can act as enhancers or suppressors. They can be in the exon or in the intron, and they direct the spliceosome as to what part of the mRNA to keep or to splice out. And the way that's regulated is by the proteins that are expressed in the cell, these SR and H and RNP proteins. Okay. This is trying to show you that you can have a mutation, a change in the gene sequence, that can change a splicing enhancer to a splicing suppressor, so that you will get, in this case, a protein without exon 7 that becomes an unstable protein. So, what type of data would you see? Well, you would be running an RNA gel, so you'd be looking at the RNA, and you'd be looking at different sizes of RNA, right? So if you have an exon, this piece of RNA is going to be larger than the RNA without that exon. And what you can see is sometimes in a cell you'll have different types of the RNA expressed. So in this case, in cell type A, you have mostly the RNA that includes exon 13, but you have a little bit of the alternative splice variant. And in cell type B, you have a little bit of the RNA that includes exon 13, but more of it is the RNA without exon 13. Okay. So to look at alternative splicing, you look at gels and you look at size difference to figure out who has the exons and who doesn't. This is just another example, and I like this example because it can show you how specific you can get. So in this case, this CAMK2D protein can express 3 different alternative splice products. Okay. In the blue here, oh, I do this wrong. Darn it. Let's see, let's just, oh, I spent all this time making these, and I know you're all not surprised that I screwed up again. Let's just delete these all. And draw them correctly. Oh, come on. All right, so blue. Let's do blue. No. Blue has all 3 exons. So blue, which you cannot see, light blue. Okay, go away. This is your blue, all 3. Okay, so these are different cell types showing which cell types produce which protein. So in SKM cell types, it's all 3 exons included. Okay, the red is where you include a little bit less of 21. And 23. 21 plus to 23. Okay, so this is this splice product. So it's a little bit shorter, you're losing this compared to 21, 22, 23. Okay, so that would be this splice product here. This is this one. And the smallest, we'll do in green, is when you connect 21 and 23 and you eliminate exon 22 altogether. So you can get a lot of data from an RNA gel that's showing you the different sizes. Right? And remember, biggest on top, smallest on the bottom. Okay, so the summary: splicing with gene expression control is controlled by these SR and HNRP proteins. They are specific for all of these splicing enhancers and splicing suppressor sequences that can be found in the exons or the introns. And they work with the general factors of the spliceosome to control alternative splicing. All right, the next way that gene expression is controlled is through mRNA degradation. And this is a natural process. And remember, we said that the cap, 5' cap, and the poly A tail are important for translation, and they're important for splicing, and they're important for stability of the RNA. But there are always proteins ready to degrade the mRNA, which makes sense because we don't want the mRNA to be around forever, or else once you turned on a gene, it would never be turned off. You would never stop making the protein. So what happens is there's an initial deadenylation. So there's a protein that will attack that poly A tail. And remember that the poly A tail normally has poly A binding proteins on it, but also remember these are dynamic. So the poly A binding proteins come on and off, and so this deadenylation enzyme can start decreasing the size of the tail. That's going to lead to instability. There are also decapping proteins that will cleave the cap and allow for 5' to 3' degradation. When you lose the tail, you get 3' to 5' degradation, and more decapping, right? This is happening primarily in what's called the P bodies. P body stands for processing bodies, and this is just an area in the cell. It's not membrane bound, it's not an organelle, it's an area. And the P bodies, in fact, here are shown in red. So you've got the nice, beautiful nucleus in blue, and then all these areas of high concentration of mRNA degradation. And what this picture is trying to show you is that as you lose that poly A tail, you start losing the interaction with the cap binding proteins. That starts making the RNA not only unstable, but you're not going to recruit the ribosomes, so you're not going to continue translation. And then eventually the RNA gets decapped and the poly A tail, and this is normal mRNA decay. And one of the slides I showed you at the beginning of this unit was that cells, different cell types have different mRNA half-life. So that's just how long is the average mRNA staying around. And this is talking about the types of different RNAs. So you can see in humans, right, the majority of our RNAs only stay around for, say, 7 to 15 minutes, and then most of them are destroyed. Yeast have a subset of mRNAs that stay around for greater than an hour. Bacteria, really quick, because they're doing rapid cell division, rapid growth. It can depend on the specific protein. So this graph is just showing you that a specific type of protein called PPR, they're plant proteins, have a much different mRNA half-life than the general transcriptome. Okay, so the transcriptome means all the mRNA that's normally transcribed. So you can see that in plants, they have quite a different mRNA half-life than humans or yeast or E. coli, and that the majority of their mRNAs are staying around for a day to 2 days, some even 3 days. This subset of proteins, their mRNA is degraded much quicker than the rest of the transcriptome. So normal mRNA half-life is normal mRNA decay is a way of regulating gene expression and not keeping those mRNAs around forever so that they continuously get translated. How is this all controlled? It can be controlled by the general mRNA degradation pathways. It can also, and this is again in plants, pretty interesting, be controlled by time of day. So other proteins that the plants are expressing in their circadian rhythm affects different RNAs. So just to understand this, there's a gene called CCL. You don't need to know what it's for, but when the plant is in the dawn mode, right? So sunrise early in the day, this CCL mRNA is pretty steadily expressed, and they've quantitated that right here. In the afternoon, this RNA is expressed in greater levels and then rapidly, or it doesn't hang around like it does at these low levels, and then it's destroyed as time goes on. So you've got a 2-hour interval. Right? Same thing happens with this SEN1 gene. It's fairly stably expressed at low levels in the mornings, and in the afternoons it is expressed at high levels and then quickly degraded within a 2-hour time point. So I show you this just so you can understand that mRNA is naturally broken down, and there's lots of factors that affect how fast an mRNA will be broken down in the cell. Okay, now we're going to switch to a brand new topic called RNA interference. And I wasn't sure we were going to get to this, but I'm really excited to show you about this. This was part of my research in plant biology. I was working on this pathway. In mammals, we're going to talk about siRNAs, and we're going to talk about it's over here. MicroRNAs and siRNAs are over here as well. Primarily in mammals, we use microRNAs to control gene expression. Plants rely heavily on both micro and siRNAs. Okay. So these are functional, non-coding RNA molecules that control gene expression. So that's why it's called RNA interference. The RNA molecules are interfering with gene expression. And you have two types of, we call these small RNAs, because they are, depending on who you talk to, 20 to 25 nucleotides in length. That's pretty dang small. Small interfering RNAs, the source, where they come from, is double-stranded RNA. And double-stranded RNA does not normally arise. It can arise from RNA viruses, and lots of plant viruses are RNA viruses. This is why plants use this as a gene control method. Humans, when we have long stretches of double-stranded RNA, we actually turn on an interferon pathway that goes and marks the cell for destruction because we know there's an infecting virus. So we don't use siRNAs very much. But how this works is you have this long strand of double-stranded RNA. There's an enzyme called dicer that cleaves, so cuts, double-stranded RNA. And so what dicer does is it cuts that double-stranded RNA up into a whole bunch of small interfering, SI stands for, oh here it is, small interfering RNAs that are going to go on and control gene expression. MicroRNAs, on the other hand, which are abbreviated mRNA, the source is the DNA. So these are genes. These are encoded genes in your genome. And as many as, they say, 30% of our genes are controlled, gene expression is controlled by microRNAs. So you have these genes, and if you look back on our RNA polymerase chart, I believe it's RNA pol 3. No, RNA pol 2? Oh, now I can't remember. Darn it. Let me look really quick. RNA, chapter 14. Sorry, I should have looked at this before. Or is it chapter 13? Thank you for your patience. Not that you have a choice. RNA. Okay, so RNA Pol 2 and RNA Pol 3 transcribe microRNAs. Okay, so they're encoded in your genome. They are transcribed and they form this hairpin structure, which is recognized by dicer. Is first recognized by drasha. So you cleave off the ends, and then the hairpin is recognized by dicer, and this is your functional microRNA. So let's talk about what those do. So here's a table from your book. It talks about the origin. So siRNAs are from mRNAs, viruses, transposons. MicroRNAs are actually transcribed on purpose from the DNA. They're both similar in size. Okay. The target is different. So the siRNA targets the RNA that it was transcribed from. MicroRNAs target other genes. They are really controlling gene expression. So let's look at this. Here we made our microRNA after dicer, or our small interfering RNAs after dicer, and they both interact with RISC. RISC is RNA interference, silencing complex, which means a bunch of proteins. And RISC can have a couple different effects. Okay. The siRNA or the microRNA, these small RNAs act as guide sequences for RISC. So they associate with the protein and they guide the RISC complex to a target RNA. Right? So they're guiding RISC to a target RNA. If they're a microRNA, they usually base pair with the 3' UTR, and they inhibit translation. And this little bump here means imperfect base pairing. So it's not 100% AUGC base pairing. They work by imperfect base pairing. They inhibit translation. I'll show you that in a second. Smaller interfering, because they came from the source. So you chopped up the source and then you're producing more of the source, mRNA or virus RNA. They have perfect base pairing, and that tells RISC to cleave and chop up the RNA. Okay. So siRNAs guide the RISC complex to the target, and the RISC complex, the protein complex, chops up, degrades, cleaves the target RNA. So you can imagine this is gene expression control because the mRNA is gone or the virus RNA is gone, and so no proteins can be made. MicroRNAs work a little bit different. They inhibit translation by physically sometimes blocking the ribosome. They're also associating with microRNAs, and this is not a great picture because it should have a little hump here. EGO1 is part of RISC. It was just the best picture I could find for translation inhibition. And a lot of times it's here in the 3' UTR. Can bind other places, but majority of microRNAs bind the 3' UTR and block translation. What this means is that sometimes, this is again like we talked about the transcription stalling, sometimes translation is restarted or allowed. So sometimes, this is not a permanent block, but it can be used as a holding pattern. And you stop translation until it's needed, and then the microRNAs and RISC leave, and translation can happen of that gene. Okay. The final way that siRNAs can work in the cell is transcriptional silencing. So siRNAs can react with a different complex called RITS, RNA interference transcriptional silencing complex, that interacts with the DNA. And causes epigenetic changes. Okay, so the siRNA associates with the RITS complex. It recruits a methylating enzyme, DNA methyltransferase. The DNA gets methylated, and you know what that means: inhibition of transcription. So RNA interference is a way that small RNAs can affect gene expression control by silencing transcription, by repressing translation, or by degrading the messenger RNA so that it can no longer be used as a template for protein expression. All right, that finalizes chapter 17.